biomarker studies with the basic science at the CTC core. Many of you may not know the CTC core, so I first give just the information where the CTC core is at the Olsen building, 8th floor. And then I know we are in the Lurie, so hopefully the CTC core can be a bridging service linking the clinical and the basic science together. As Sarah said, my name is Huiping Liu. I'm an associate professor in pharmacology, and I've joined the appointment in medicine and HEMOC. Here is just showing again the history, how the CTC core is established. So it started by Massimo, who had a CTC lab at the Olsen 8th floor in 2016. So over 7 years, he actually built this facility until he left in 2021. So I took over after he left. And then now still have Chan managing the core, and the Huiping is running all the CTC instruments. Basically, Huiping is here today. He's introducing some of the instruments at the end of my talk. And I appreciated the clinical collaborators, especially Dr. Bill Gradishaw, and then also Janice Liu, who just joined us this September, and she's also in the audience. So representing the clinicians or physician scientists who can help us link the clinical research with our basic science. I also wanted to introduce, since this is our breast cancer research program, I just wanted to introduce you our planning committee. We had the meeting a few weeks ago, and to start our monthly seminars, and then including basic scientists, clinicians, as well as a lot of technology experts, and also epidemiologists and who are doing population science. So we had a first planning meeting. So today, I really appreciate it. We have this first seminar starting. I wanted to really highlight our program manager, Gina, who is also here in the audience. And then if any of you wants to join us, please just contact me or Gina. So we would like to have you join our program, and we hope to build our website again and adding more scientists from clinic side, basic science, and population science as well. So this is my just the summary how I come to this the CTC service that helps my basic science development, and then we published a few papers since the collaboration. When I moved here in 2016, I had this discovery of we found a cluster of floating clump of cells that is a circulating tumor cell in a PDX model that carrying triple negative breast cancer in humans. And this is a circulating tumor cell cluster floating in the vasculature. However, we didn't have a enough clinical data. And then, but we collaborated with Massimo to continue the CTC studies that actually a lot of those published from the last few years, and I'll just summarize here and then give more details later. So the basic discovery, I just want to summarize. We identified many of the surface proteins, including CD44, that is a cancer stem cell marker, and the CD81 is a partner of CD44. And also the ICON-1 later on mediating the circulating tumor cell cluster formation, and then poly-XL is a chemo resistance related molecule, also upregulated in CTCs and CTC clusters. And most recently, we identified plexin B2. I know a lot of surface proteins identified that are enriched in the circulating tumor cells as well as circulating tumor cell clusters. And then functionally, they contributing to tumor cell aggregation and then shedding into the vasculature or blood vessels, and then circulate with or without immune cells to promote metastases of breast cancer. And from there, we published a few papers since 2019. I'll summarize 4 stories later. And then as part of the liquid biopsy, and then when we collect the blood, we also isolate plasma, and then exosomes can be identified or detected in the liquid biopsies, and a lot of the exosomes secreted by cancer stem cells can also promote stemness of tumor cells and then promote metastases. After all of the basic science discovery, we wanted to develop antibodies to block CTC cluster formation, and including a lot of antibody and exosome engineering now is going in my lab. I'll touch a little bit later on how we actually use machine learning approach. This is a part of Hannah's project, who's also here. And then we recently submitted you will want to look at the 3D spatial structure and the tumor-immune interactions in breast cancer. But I really want to highlight all of our research has been facilitated by the CDC core, and especially the FDA-approved cell search that has collected over 400 patients' blood samples and CTC images. I know Huiping is going to talk later. I just want to give you a highlight, a summary of all the instrument at the CDC core that includes the FDA approved cell search system. This is the most used or most commonly cited in our research, and that is also the first FDA approved instrument to use the immunofluorescence staining to find the CTC cluster, CTCs and the CTC clusters. The second instrument FDA approved is Biomarkers, that is non-specific to cell surface markers, but based on cell size. You can choose the pore size like 8 micrometer or 10 micrometer. Any cells bigger than that will be collected. And a lot of them will have enriched the CTCs because epithelial tumor cells are bigger than blood cells. The other instrument, that array is currently is a rent instrument they can use for single cell capture and isolation, and then also fluorescence microscope and digital PCR used for DNA or mutation analysis. And we have a wish list, and we have been putting grants together. If you wanted us to include any other instrument, we hope to expand the list, including image stream, and then a cell selector is a robotic-based cell transporter, and then or capturing. And then we would like to also collaborate with Bill Mueller to set up intravital imaging in the future. And then we have a potential instrument to include is the exosome single vesicle analyzer Apogee. I just wanted to highlight a few stories. As I mentioned, when I moved here, even we discovered this phenomenon of CTC clusters in breast cancer. This is a PDX model or a mouse tumor model in using the intravital imaging approach in collaboration with John Condelis and Albert Einstein. So this is a new mechanism. I know by default, by literature, you may have seen single cell mediated dissemination of metastases or single CTCs. However, we do observe the CTCs interact with each other on top of vasculature or within the vasculature using intravital imaging approach. I know many of you may not believe CTCs, but you don't believe because you don't see it. If you see, you'll see circulating tumor cells do exist, and then this is the most vivid evidence using the intravital imaging approach. And many of those interact with each other or forming clusters on top of vasculature before they get into the blood vessel. And then also the others can actually circulate as like loose clusters within the vasculature. And I'll show that later. But even with this discovery and a lot of basic science, mechanistic work, like CD44 would mediate the cluster formation. Our paper initially in 2017 was rejected by Science or Nature Cell Biology without much clinical evidence. So that's how we get to collaborate with Massimo in 2017. Once I moved here, there was like a perfect collaboration. So Massimo and Huiping helped us to compare the single CTCs and the cluster CTCs from breast cancer patients. So that's why you see it because you believe it because you see it. Now the clusters are definitely existing in the human blood, and then this can be shown by multiple approach. The cell search is just one of the instrument or methods we use. And we have also used other approach to complement the blood processing if there are any artificial effects. So we use the IHC, for example, staining from human patients. If you have never seen CTC or you don't believe, you'll see that in tissue sections in the vasculature structure. And here you can see a cluster of tumor cells do loosely attached to each other. And then these same phenomenon we have observed in patient-derived xenograft models in mice. This is the lung metastases of the mice. So this is a patient with triple-negative breast cancer also in the lung metastases. And then you'll see this represented in PDX models as well. But the blood processing can confirm this, and using Massimo's clinical data, we have also observed a lot of the CTC clusters, and we have also observed that using the PDX models, this is also isolated from blood. You'll see tomato labeled tumor cells actually forming clusters as well. So these are the evidences we collected in both human and mice. All right, and then in collaboration with Massimo, we have been able to compare the instance of single cell and the clusters in human and PDX models. So they are very comparable. And also here, this is our 2019 cancer discovery paper to look at CD44 expression in CTCs and in both vascular, we call the vascular CTCs. If we look at using IHC method, that does not have any non-specific processing artifacts, and then the blood processing may cause some artificial effects, but both evidence support the enriched CD44 expression. And the other markers we have identified later on, like plexin B2, CD81, ICON1 in the clusters comparing to single CTCs. And then more importantly, with the cell search of the CTC core that look at more than 100 or 400 patients and then over the years, and then we compared the patient survival that had some of the patients would have a CTC cluster detection and compared to others who don't or only have singles, or non-CTCs, that the patients with detected CTC clusters would have unfavorable or worse overall survival compared to the other group. And then especially even for CD44, only we looked at a few patients. The patients had a CD44 positive CTC clusters would have also worse survival compared to the other patients who only have CD44 negative or non-CTCs. So this helped us to publish our paper in Cancer Discovery in 2019. This is the first story including the other functional studies basic scientists will do, because if you knock out CD44, the tumor cells will not cluster and not metastasize the lungs. This is what we did. If we combine the red and green tumor cells, one is wild type, one is knockout, you'll see the knockout tumor cells will not form clusters or colonize in the lungs. This is evidence from the functional studies and will support the importance of CD44 in cluster formation and metastases. I'll skip the summary because this is similar to the other proteins. Or you have seen that in the beginning. But I wanted to highlight the second story we just published in Cancer Discovery again this year. This is led by Newman. That's the WAG in the lab, and we collaborated with Dr. Bill Gradisha and the other physician scientists or breast oncologists like Amy Shaw. This is a new discovery from the clinical side to the basic science. So we have been interested in clusters, but we have not linked it to the treatment before until to this paper. Massimo's work initially also helped this in the beginning, but the ongoing studies were helped by Dr. Gradisha and Amy Shaw. So this is a discovery. We're looking at how single cell CTCs or clustered CTCs longitudinally change before discovery and before therapy or after therapy. A few years ago, the standard of care for triple-negative breast cancer is chemo, although later on, since the pembrolizumab, now the standard of care will change or be updated. I'll cover a little bit later, but chemotherapy was the standard of care before. So we looked at the patients from the last few years with chemo or without chemo. Some of the patients, the single cell CTCs with chemo or without chemo, before treatment and after treatment, these are 2 time points. We didn't see significant change after looking about 40 patients. However, the clusters, CTC clusters would change more for the patients who had chemotherapy. You can look at it here, from here, the lines. The blue lines are the patient longitudinal tests at the baseline before chemo, and then 3 months later after the first cycle of the chemotherapy treatment. You'll see a lot of blue lines increase for the CTC clusters, and then we have seen the significant increase the CTC clusters for some of the patients had chemo treatment. I know chemo has been beneficial for some of the patients, but not for all of them. For those of the patients who don't respond to chemo well, they may have increased the CTC clusters, may be responsible for the worst prognosis and even increase the metastasis in those patients. So like if the chemotherapy doesn't kill tumor cells or make tumor cells stronger, so that's really bad outcome. And you'll see also longitudinal tests of CTCs will be correlating with the outcome of the patients. So we stratify the patients based on the CTC change. If the CTCs increase, even including the singles or the clusters, those patients are usually come together. When the single cells increase or when the clusters increase, they always sometimes correlate with each other, however, not all the case. But increase the CTC detection after chemotherapy correlated the worst outcome of these patients. If the CTC numbers don't change, they actually have an okay outcome. If the CTC numbers even decrease, it's a great biomarker to suggest that patients will have the best overall survival. So this is a great biomarker used for clinical probably prediction of those patient outcome of the or therapy response. I know pathological complete response has been a standard or the gold standard for response assessment in the clinic, but a CTC cluster or single CTC change the numbers alterated longitudinal pattern can be used for biomarker as well. So this is a great clinical kind of biomarker discovery platform. And then even there, we compared the chemo receiving group and the chemo non-receiving group. You'll see the chemo receiving group never had maybe this is small, still a small size of patients, but we have only seen 2 groups in the chemo treated. Either they don't change or they actually become worse. And however, if some of the patients didn't receive chemo, they could have still decreased the CTCs of the therapy. So This is a very preliminary work. However, we wanted to combine with our basic discovery. So we look at the surface protein change as well as the glycosylation pattern change. So this is what Newman discovered from looking at all the glycan residues or the patterns. And she identified the cellulation pattern actually changed in the clusters, especially after chemotherapy. And you can look at it here. The clusters would have a sialic acid low and compared to single CTCs. And then this one is after looking at thousands of CTCs from hundreds of patients, you'll still see this same pattern and significantly change in the clusters. And these clusters, I'll try to move this away. Yeah, have been showing associated with chemo treatment more significantly. And you'll see with the chemo treatment group, you'll see more clusters with decreased cellulation pattern change. And the cellulation is a sialic acid with negative charge that may actually exclude the CTCs from cluster formation, and decrease the cellulation would make the tumor cells cluster together. So this is a ongoing basic science study by Newman. So she demonstrated in the PDX models or mouse tumor models, if you treat with the sublethal dose or like a suboptimal dose with paclitaxel that doesn't shrink the tumors that resist to the paclitaxel, then the tumors will have shown increased metastasis to the lungs. So paclitaxel, if it does not kill the tumor cells or the tumor cells resist the paclitaxel, they will actually develop more metastases. And this is associated with the sialic pattern change, and the paclitaxel will actually increase the CTC cluster formation in the mice, and showing the also cellulation pattern changed in the clusters, and also paclitaxel treated mice. So this is a discovery from the clinical side and extended it to a basic science. And then Newman went on to look at the features of the CTC clusters, whether they are associated with chemo resistance or chemo evasion. So she observed from the CTC, the cell search analysis, the Ki67 staining pattern will be actually positively associated with the DAPI stained DNA content. Then you will see a perfect kind of correlation.
If that's okay.
Whenever the
Yeah, I can send you the Zoom link.
is
so you
higher
can see it.
or more intense in the clusters or the single CTCs, you also see
Problem.
Ki67 expression. This is actually showing in the clusters would have more quiescent pattern, and these are showing 5 CTC clusters. Only one had a Ki67 staining, and this one had a little bit higher DAPI staining as well. The others negative for Ki67 would be actually weak for DAPI signal. So then we use the DAPI signal to serve as a proliferation index, and you'll see the R associated with the cell cycles. This is what I demonstrated by Newman. The DAPI staining will actually showing a correlation with the G1, G0 at rest, and then also other cells will be actually in G2S phase. So this is a very useful also correlation for biomarker studies. Our project number 3 is to look at tumor immune cell interactions. We had these discoveries from our CTC derived models in mice. We compared the primary tumor cells and the lung mass. We have seen primary tumor cells actually, or the lung metastases would have changed the proteins and then forming the immune signature. However, we don't know what is going on between a tumor cell and the CTCs, between the immune cell and the CTCs in the blood. So we wanted to identify drivers to regulate CTC immune cell interactions. So This is just showing you the evidence again, as I mentioned. This is the mouse tumor model. It's a poly-MT model in mice, and then the monocytes is white here, is having the blue FP, but showing white in the red background. You'll see this is a snapshot of 3 tumor cells escorted by 2 immune cells, and it's showing here as intravital imaging. This is the evidence the immune cells or white blood cells can interact with tumor clusters in metastases. And also by human blood analysis, this is done by David in my lab. So he has looked at all the immune cells in the clusters of CTCs that would have immune cells. We have observed most of the immune cells would be myeloid cells, including neutrophils and monocytes, but there are other rare T cell populations there as well. So today I'm going to focus on another project. We look at monocyte interactions with CTCs. So we did a proteomic ranking of all the breast tumor proteomic analysis using mass spec and all the cell lines, including the CTCs, as well from the patient or from PDX. And we have done the computational ranking to look at what proteins, especially surface proteins, or be most abundant in all the databases, including primary tumors, CTCs, cell lines, and also more specific in tumor tissue versus normal. So we identified Plexin B2 as one of the top, especially it's associated with patient overall survival or distant metastasis-free survival. And we have done also the tumor tissue array. We have seen the Plexin B2 as highly expressed in triple-negative breast cancer and also other subtypes. And then more importantly, we also have used the cell search approach, though not including here, enriched expression in CTC clusters, more specifically enriched in CTC white blood cell, the heterotypic clusters. So we went on to look at the mechanism of Plexin B2 mediated interactions for tumor cell and monocytes. We used the THP1 for functional studies and also used the mouse monocyte interaction with 41s. We have observed the knockdown or knockout. Plexin B2 would actually compromise the cluster formation with the monocytes. And then the monocyte 4A, the simagraphy 4A would be the ligand of Plexin B to mediate the white blood cell or monocyte CTC interactions. And this paper is under review right now. We have also done mouse studies when we knock out the Plexin B2, the tumor burden didn't change or still comparable. However, the lung metastasis is also gone. So we are developing anti-Plexin B2 as a therapeutic approach to block in the coming studies. So this is just showing you also using the CTC core, and then we have been able to demonstrate the CTC interactions with immune cells, especially one of the cell types is monocyte, and then we have other upcoming studies by David showing the T cells will also be interacting with CTCs. So this is just to summarize again how we use the CTC core to help our basic science, or how we discovered some of the phenomenon from the clinical CTCs and then come to understand the mechanisms of CTC cluster formation with the homotypic CTC cluster formation, as well as the heterotypic CTC immune cell interactions, and both would promote metastasis for breast cancer. And then in the meantime, whenever they cluster together, they can also promote stemness program, especially like a CD44 mediated upregulation of OCT4 and then FAC signaling pathways. And we are developing a therapeutic approach to accomplish the therapeutic blocking of CTC cluster formation. This is just showing you a few examples. Antibodies are definitely our next step candidates to block CTC cluster formation, and we have many surface proteins as targets. The challenge, of course, will be making it tumor specific because many of the proteins may also be expressed in normal cells. So that's how the glycosylation or post translational modification patterns may matter, because those can be tumor specific, like glycosylation patterns. And many of these proteins are glycoproteins. So we hope to identify tumor specific glyco epitopes of these proteins and develop antibody development or therapeutic blocking strategies that can further activate immune cells for cytotoxic killing of tumor cells. Another story I want to highlight based on the CTC core productivity. I know Huiping may cover this more later, but I just want to highlight in the CTC core from the last 7 years, how many samples. There are 3,000 biopsy tests have been conducted at the core, and many of those are breast cancer specimens, but there are many others, colon cancer, as well as other disease. But these are the biobank of liquid biopsies we can use not only for CTC analysis, but also for any plasma or serum component analysis. I know Dai has some projects wanting to look at the plasma or the liquid biopsy components. So definitely those are useful for all the basic scientists. And then just want to highlight the CTC core is the only core in Illinois or Chicago. Right now, we hope to be able to get a clinical license. This is ongoing effort. So maybe get a clinical certified service for reimbursed analysis of clinical samples. I want to highlight another project we just started this year in collaboration with computational experts, and then especially artificial intelligence experts like Carson Stranger at HHMI and Yuan Lu here at the Northwestern. Since a lot of CTC images we collected, maybe over millions of images we collected, we wanted to use the AI approach for automated like segmentation and the CTC feature analysis. This is just at the beginning, but I wanted to show you some progress. And Josh in my group and then Huiping have been collaborating and with the help of the other groups, Andrew, David, and Chen Shen in Yuanlu's group. Each CTC analysis on the cell search platforms scans at 175 frames of the cells actually captured by the CTC platform. There are maybe thousands of cells captured, however, only maybe a few CTC cells or up to hundreds. How can we recognize the CTCs? A lot of times Huiping has to go through that manually to verify the CTCs. However, we wanted to use the AI to help us to identify the features of CTCs as well as the CTC interacting immune cells. I know artificial intelligence can be very transformative. This is only at the beginning to harness the power, and then we hope to extend from the CTC discoveries to eventually drug discovery, and then also using the AI approach for protein structure analysis and the drug development. And this is just showing one example. I have been collaborating with Lurie and Yang Shen. Yang Shen is a computational scientist. Lurie is an immunologist on campus. And then we have been developing kind of AI or language model-based antibody structure analysis, interaction with any antigens, although we used it in COVID first, but we hope to expand to cancer discovery and also the cancer antigens later on. This is just showing you the principles from thousands of antibodies with sequence from human B cells. So we can use this program, the machine learning program, or the language model, the models, and then the physics-driven or data-driven approach to optimize the antibody screening, and also even for redesigning. And this is the study being led by Hannah in my group, and then we hope to extend it to many of the cancer target therapeutic development. So I wanted to, since this is a breast cancer research program seminar, I just want to highlight again our thematic topics, and we wanted to highlight the breast cancer program at Northwestern, really unique to our school. And we want to highlight the potential immunotherapeutic development in triple-negative breast cancer, or the tumor vaccines or any other nanomedicine can be developed on the campus. I know many of you might have interest in immunotherapy, and we hope to be able to submit a PO1 or SPORE in the future, maybe from some of the thematic topics. So the other thematic topic is metastases and therapy resistance, as I just covered circulating tumor cells and cancer stem cells. So there are multiple studies on campus, and we hope to be able to consider a PO1 submission. Again, many of our studies, we wanted to use advanced technologies, including computational intelligence, big data, and then also the ongoing work using spatial 3D multi-omic mapping. That is our UO1 project. And then a lot of studies, including Simon Khan or other population scientists, are related to breast cancer prevention, including the incidence prevention or recurrence prevention. And then another topic in our program is stress response, like from ROS or ribosomal translation, that can be also collaborated projects together for maybe a PO1 submission as well. I want to highlight we have preliminary discoveries, especially in triple-negative breast cancer, comparing Black patients with white patients. We have seen increased mortality rates in Black. That's why we want to compare an African Americans versus European Americans and see whether the tumor immune features might be different. These are our breast cancer research program. Our initiatives, including we quarterly strategic planning meetings and monthly research seminars. So this is our first one. We hope you sign up for the next year, starting January. I think January have two spots filled, but February we still have one to be filled. And then we are planning annual breast cancer research symposium in connection with LINSAGE. And then we wanted to do a collaborative project submissions, including UO1, PO1, or SPORE. Although the SPORE we may do 2024 as well, we are still planning. And if any of you are interested in joining, please let us know. We hope to be able to collaborate with the clinicians, especially Johnny's and then Bill Grady-Shar for new clinical trials, and that is required for our SPORE. And then Gina has been launching the website, and then we would have training programs as well. So if any of the trainees, graduate students, postdocs, or staff, you want to join us, please let us know. We can add you the email, the list. And these are the NCI, some of the RFAs, but many of those you may have seen, but we wanted to highlight a few the investigator-initiated trials. and also some of the liquid biopsy related actually R01s you may consider. I have also heard at the San Antonio meeting there is a disparity, actually a study section you probably never heard. I think many of us, if you want to work on triple negative breast cancer, it's possible to also submit it to that study section, and that is less maybe competitive compared to others, especially if you include both population science and a lot of basic science, it may be a stretch. So this is a potential PO1 or spore, although Derry and I discussed we might have this 2 projects now, but we are looking for a 3rd project to do a spore or PO1 together. If you have any drug can be proposed for IND enabling and the phase 1 trials in the next 5 years, so you can join us for this PO1 or breast spore submission. I think I wanted to highlight our monthly seminars. Please contact Gina to sign up, and then we still have the February spot open here, and then starting January 12, and then we'll have these monthly seminars. I'll probably stop here and answer any questions. This is a highlight again. I want to thank my lab who contributed a lot of work, and then my collaborators, and then these are the over years, the lab members now, and yeah, thank you. Any questions?
We have 2 actually online. So do you have any comments on intracellular bacterial pathogens in metastatic breast cancer, which drugs include them?
Can you repeat again?
So Mark Cannon is asking whether you have any comments on intracellular bacterial pathogens in metastatic breast cancer.
That's a great question. We have not looked closely, but maybe we should revisit the RNA sequencing data, and then eventually we'll see if there are bacteria components or the other pathogens. But inflammation is certainly involved. Maybe some virus infection could be, but we have not closely looked at. But that's a great question.
The second question from is there any correlation with ctDNA and the CTC level?
Yes. I didn't talk a lot of circulating tumor or cell-free DNA, but there are a lot of studies that even in all of our breast cancer patients, many of those we had a cell-free DNA analysis, and many of those had mutations. I would say in ER positive tumors or HER2 positive tumors, they are ER like mutations actually detected, and then you may show that data later. And then those are associated with CTC detections, especially for therapy resistant patients. They would have the CTCs detected with the mutations, and it could be corresponding to the cell-free DNA mutation analysis too. But cell-free DNA is also detected from killed CTCs. So not only from CTCs, but from some dead cells too. So cell-free DNA may give you even more mutations than the circulating tumor cells. So it may be more accurate to find the mutations in circulating tumor cells that are the real targets. However, cell-free DNA, it also changes longitudinally. If it's immediately after chemotherapy, you may see actually increased cell-free DNA. However, for stable patients, you may see decreased after the first peak. Yeah.
For the CTC, do you know like which factor contribute to the CTC cluster, negative or positive? Another question like the single CTC versus the cluster. So do you know which factor?
There are many factors like we discovered that like chemotherapy can be associated with the CTC cluster detection. I'm sure the immune fitness of patients will be certainly, because the CTC clusters actually, when we did a basic experiment, they are more sensitive to immunotherapy. For example, why the CTC clusters detected in those patients, but maybe because the immune system is very weak. So they are actually can circulate without being killed, means their immune defense system is also very weak.
So they're relevant to the type of the cancer. So
we have seen the cell search detected mostly the epithelial tumor cells, but all the metastatic cancers presumably have CTCs because they have to spread from blood or from lymphatics, right? So you would see they should exist in all cancer types, but the current detection approach, like a cell search, is focusing on epithelial tumor cell markers. So we hope to expand using the flow cytometry, for example. If we can sequence all the CD45 negative cells, those should be having enriched CTCs, but not all the CD45 negative cells are CTCs. They are just enriched in CD45 negative population, especially the single CTCs. However, we have also observed a lot of heterotypic CTC immune cell interactions that cannot be probably detected sometimes in CD45 negative cells because they would have some another cell, CD45 positive. So we have been looking at CD45 positive, but epithelial positive, like double positives as a heterotypic cluster as the marker detection. But in single cells, CTCs should be enriched in CD45 negative cell population.
Is there a way you can block the CTC omitt?
Yeah, that's how we wanted to develop antibodies to block the CTC cluster formation. Yeah, so that would be the goal. Yeah, next step.
You observe in like heterogeneity in the CTC, like in the PDC cluster,
That's a great question. We always wanted to expand to epigenetic analysis, but we have only looked at mostly the protein level to begin with, but definitely expand to a transcriptome and maybe translational control can be also splicing. We have seen splicing change as well in some of the CTCs. Anything related to stemness regulation can be related to cluster formation because we have seen clusters enriched with cancer stem cells. So any stemness related programming will be certainly related. Yumin, you wanted to, cover a little bit more the CTC core.
Good afternoon. Little bit nervous. Don't kill me. I'll present better. Just the dot here. Okay. Yes, I work with Chang and at a core facility, CTC facility, circulating tumor cell facility, and closely with my boss's work, doctor's work. And all missing is easy to understand. That means right now the precision medicines is so hard on clinical trials. And for us, we want to be helpful to sort out the circulating tumor cells, self-readings, do the relationship analysis. And the CTC enumeration, isolation, and check the recovery of single cells, and the marker epitope independent enrichment of the CTCs, immunofluorescence, microscope, GDPCR, whole genomic genome amplification. The reason I put here is for here. I'm missing this. This we are working on that, but not a lot, but still have some publications here. And Dr. Liu mentioned about the CTC cell search, and this machine has been used around 20 years. And so right now, even we have some other sorting machines or instruments or systems, but always compared to this one, because this one is only the FDA approved. That's the reason why it's so consistent, very consistent. I feel, very comfortable to work on that completely. We tried, when I joined Dr. Liu and Dr. Rufani's lab, and we worked on several systems for the CTC processing or isolations. But right now, look like this one is a better one. We keep this one all the time. Others just like the helpful one. And besides the CTCs, after another one is debris. That means most of the samples we got from the cell search is pool cell. That means lots of cells mixed or background cells. But if we want the single cells analysis, we go to the debris and to recover the single cells and then do the next step for the deep analysis. And besides this, cell search and debris, we have the prosthetics one. Prosthetics one, that means we do the live cells analysis. That means you get the blood from the patients, then go to these machines from the channels, made the cells, separate them depending on the size. To get rid of to get rid of the red blood cells, white blood cells, to keep the bigger ones, tumor cells at behind. Then we recover that and do some next step. And fluorescence microscope, everyone knows. Another one is ddPCR. That means the PCR's cocktail. We make droplets first, then make the PCR, then analyze all the droplets signals and get doors. Whatever doors we have there, maybe, I'll show you later. Oh, you copied that. Yeah. And this picture is cell search cells. Dr. Liu showed you most of the CTCs like this only, the single cells or the clusters. But here I show you some white blood cells bonded to the CTCs. That means the story more complicated. This kind of CTC clusters, we claim them hetero clusters. That means different kind of white blood cells can be associated with CTCs. Why they aggregate and what does that mean? Was a relationship to be impacted to the patient's prognosis or some things? That's the current story we want to develop. And David and Josh and Andrew and in Dr. Liu's lab, we work together on this. Some people noticed that, for us, we did a lot. Dr. Liu mentioned a lot of the CTC clusters formation or some things, the mechanism study. But other people mentioned about the white blood cells, the neutrophils can put the CTCs growing proliferation worse and bad prognosis, and some T lymphocytes and the killer cells can kill the tumor cells directly, and even the macrophage cells can bond with CTCs and to form the double positive CTC cluster with fusion, to be the fusion cells, or separately. What does that mean? It's a big story we want to develop. Okay. And I know Dr. Liu's we work on the breast cancer. Breast cancer very common for us, but also the prostate cancer and the colon cancer and the lung cancer. And even I noticed Alice and others here, we worked with multiple myeloma a lot. That means these cell search can work with different kind of tumor cells processing. And COVID. So familiar to this. We use the cell search to process this to get a lot of the endothelial cells and ACE. These are cells debris. And the debris machines, the copies from cell search and drop in the chip and work process that and define the CTCs, and then we cover the single cells. Very fancy, I feel. Prosthetics. I can't stop to talk. Yeah, look like the blood cells, yeah, the blood goes here, yeah, channels, and then they keep the tumor cells back, and then we can collect. And this DDPCR, that means the DDPCR machine. Yeah, this is a fancy work we did, and got the ESR1 mutation. The mutation is pure mutation and combined mutations and the single the normal single Y type and the droplet and to make sure the PCR is working. Yeah, the service we did this year's really get more and more. Yeah, sometimes every day I can process them more than 10 samples. Sometimes I made 16 samples or more than 16 samples. And yeah, but very busy and very exciting. I believe, yeah, Dr. Liu give us lots of the breast cancer work, but how about the lung cancer or the prostate cancer, colon cancer, or the myeloma or other leukemia subs? I believe, yeah, if you are interested, I can be helpful. That's what we are doing.
You want to also highlight that most blood can also be processed on the cells.
Yeah. For the mouse tumor cell, mouse cell samples, mouse blood is so tiny. So whenever we process the mouse sample blood, we can donate some blood and to edit to put the tumor cell to blood to volumes like 7.5 ml, and yeah. So I want to show you this one. This is a prostate cancer. This one, the morphology is so good. Look like our finger. Yeah, apple cream metaplanar differentiation. That means the fixing solutions, the processing, keep the cells morphology is, yeah, pretty good, I feel. Yeah, I want to, yeah, this patient is the prostate cancer. Yeah.
Right,
Thank you.
Thank you.
Thank you so much. Thank you.
Great presentation, both of you. Huiping, so I want to ask you, do you have the correlation of CPC clusters versus PSL mutation, and how many samples do you run?
We have a lot of the ESR mutation analysis from, I think we have all the clinical sample with cell-free DNA analysis, so we can look back. I just didn't check how many would have, but it's a good proportion of those luminal breast cancer would have ESR mutations.
That's great. Is there a correlation of more CPC clusters with PSL mutation at all?
Yeah, we haven't looked as closely. I don't know. Christopher Nini's group might have looked, so we can check. Yeah. It's a great question.
Thank you.
And whoever designed in, could I ask you to please sign in?